A historical summary of the development of reproductive cloning. The first study of cloning took place in 1885. In 1902, German scientists created a set of twin salamanders by dividing an embryo into two separate viable embryos. With the arrival of recombinant DNA technology, it became possible for scientists to create clones with genomes containing pieces of DNA from other organisms. Dolly the sheep was the first to be cloned using the process of somatic cell nuclear transfer, causing significant changes in the reproductive cloning techniques. There are many purposes of reproductive cloning, such as for livestock, cloning animals that are going extinct, to finding a solution for infertile parents, and for people who need transplants, and cloning someone who died to balance and limit grief. How reproductive cloning works. So first, scientists take DNA out of the cell from the original animal that they want to clone, and then they take an egg from another animal and remove that egg's DNA. Then the original animal's DNA is put into the empty egg, and then the egg develops into an embryo. Lastly, the embryo is placed in a female animal who carries the clone until it is ready to be born. This is a diagram that explains human reproductive cloning. The connection of cloning and DNA, protein synthesis, or any other cellular process. So reproductive cloning requires identical sets of nuclear genetic material, DNA. Without it, reproductive cloning is not possible. Another interesting form of cloning is DNA cloning. DNA cloning is making many identical copies of a piece of DNA, such as a gene. How it works is that a target gene is inserted into a piece of DNA called a plasmid. The plasmid is introduced into bacteria through a method called transformation, and bacteria carrying the plasmid are picked using antibiotics. Bacteria with the right plasmid are used in order to create more plasmid DNA and at times also make protein. The social benefits of the issue are that animal cloning allows faster reproduction of their most productive livestock. It will eliminate infertility so everyone is capable of having their own child. Parents who lost a child could reduce their grief by cloning another child. People can treat those that are sick through organ transplant. The research on cloning has come a long way. In the past, Dolly the sheep was the first mammal cloned, which led to the discussion of cloning humans, whether it was possible or not. In the present, research is still being done. However, everyone knows the risks of cloning, and as of now, cloning humans is banned everywhere as it is way too dangerous. While in the future, I believe that there's a possibility that cloning humans would be permitted in most countries, and people would undergo cloning procedures for personal reasons, like designing their babies, or to profit off of clones that will only lead to a corrupt and broken society and threatening genetic diversity and evolution. Some cons of cloning are that the clone will have no sense of individuality as it will not be given the same treatment in society. It is a dangerous procedure as there have been many clones that have died due to health complications. Designer babies will also be created, which can be really detrimental to the clone psychologically. Some pros of cloning are that it will help eliminate infertility. However, some people disagree because they say that the procedure of cloning is an unnatural process. Secondly, another pro is that it will cure genetic diseases, but some people feel that it's not fully safe enough and the technology is not developed enough. Lastly, organ replacement is another pro, but some believe that it's unethical as it can violate a clone's human rights. Some ethical issues are that there is a high risk associated with reproductive cloning. Cloning could be used for the wrong reasons, like to breed better humans. And lastly, it goes against the natural way of life. In Canada, it is illegal to create a human clone whether the purpose is for therapeutic or reproductive cloning. It is not permitted. Over 30 other countries ban human cloning, like Canada, while 15 other countries have banned human cloning but allow therapeutic cloning and the research. My personal stance on reproductive cloning is that it is wrong. Due to the risks being too high, it is a dangerous procedure. The only type of cloning I think research should continue in is therapeutic cloning. Therapeutic cloning is really important and has the potential to save many lives with a high success rate. We can further understand certain diseases and develop new treatments with therapeutic cloning, which could lead to finding many cures.